Thanks very much, Anna. So, as Anna said, I'm a curator. My name's Paul Denny, and I work as part of the UniProt content team. And today I'm going to talk about the UniProt knowledge base, focusing on examples of proteins that have been annotated for a role in antimicrobial resistance. And of course, after the webinar's over, we'd also appreciate any feedback from you on other proteins which you think we should prioritize in future. So I'm going to go through different phases, starting off talking a bit about antimicrobial resistance. As a reminder, I'm sure many or all of you may know this already, and probably better than me. Then introduce UniProt, the resource, and talk about taking an example of a protein which is involved in AMR as one. I'll guide you through what it looks like in UniProt and what functional data there are for that protein, and then how you can explore that and also other data associated with a protein entry. Then after that, I'll talk about how you can take a sequence from UniProt and use the analysis tools which we supply to do things like blast searches, multiple sequence alignments, and so on. Then finally, the last two steps, I'll talk a bit about some examples of proteins which indirectly play a role in AMR. And then I'll say a little about how UniProt adds more value to researchers who are studying antimicrobial resistance. So AMR, as I probably don't need to say, it's one of the world's most urgent public health problems. And back in 2019, figures from the CDC and the WHO suggest that over 1.2 million people worldwide died as a result of antimicrobial resistance. And an even bigger number, nearly 5 million, died of conditions which were associated with AMR. And perhaps unsurprisingly, the highest rates of AMR are found in low and middle income countries. Something which some people may not have considered is not only is there a disease burden, it's of course a worldwide financial burden as well. The World Bank estimates that about over 3%, nearly 4% of global GDP could be lost to this condition, to the effects of AMR by 2050. Which organisms are important? Well, the WHO has got a priority list. Public enemies number one, E. coli, Staph aureus, Klebsiella pneumoniae, and excuse me if I mangle the Latin names of many or any of these species, Acinetobacter baumannii and Pseudomonas aeruginosa. And then a bigger group, the Enterobacteriaceae, which includes a number of species. Those are what WHO considers the most important, and there's this acronym ESKAPE, which I've highlighted, which is a way of remembering the species. But in addition to those, other organisms are prone to AMR. There's a list here, one of which I'll come back to a little bit later on. Moving on to talking about UniProt itself, to the knowledge base resource which I'm part of. So UniProt is a protein function and sequence knowledge base. So it holds information about both those things. So for example, is a protein an enzyme? Where is it in the cell? Is there a 3D structure associated with it? And other information such as protein-protein interaction data. So all of our data is freely available. It's updated every 8 weeks, approximately. And I've mentioned that we supply tools for sequence analysis of proteins, something which sometimes gets forgotten, but actually is at least as important, is that all of our resources have multiple links, cross-links to other resources, over 180 different possible websites and databases. In addition to those, I'm going to say a little bit about how the data gets into UniProt, and then point out that the proteins in UniProt are divided up into 2 broad categories: those which have only been through an automatic annotation pipeline and get called translated EMBL, tremble, and then another subset, a smaller number, of proteins which belong to a group called SwissProt, and they've been manually curated by expert curators. And we're dipping our toes into the machine learning and AI, the use of those tools in UniProt. So proteins which in the past would have been called uncharacterized have been named with some supervision from manual curation, but named by an AI tool developed by a Google offshoot called ProtNLM. All of the data in UniProt is accessible by API programmatically, and we encourage contributions from the community. So I mentioned just in passing there where the protein sequences in UniProt come from. The vast majority start life as nucleotide sequences in the public databases, and they get submitted to databases such as ENA, also NCBI and the DDBJ. And those then get things like open reading frames annotated, but very little else. And then they're imported into UniProt. But we do have protein sequences and nucleotide sequences, which are from other sources, so direct protein sequencing, which some labs do. PDB, the structure database, we get data from them, and also from the genome browser Ensemble. And all of this data gets amalgamated together, and some of the redundancy is taken out of it. But the tremble section of the database consists of protein sequences which are described as unreviewed. When they've been annotated by a curator, they get promoted to reviewed, and they join the SwissProt section of the knowledge base. Going into a little bit more detail, I've talked about external sources. One of the databases that I hadn't mentioned there, which underlies all the others, is an unannotated protein archive called UniPark. So this is the raw protein sequence as it comes in from these other sources without any annotation. This then gets passed through 2 automated systems, and I'll say a bit more about those in a mo. And a subset of those then, after being automatically annotated, get annotated by an expert curator. And so that involves using the literature primarily to add data, functional information about proteins to those entries. And the great beauty of this is it's a kind of cyclic process. So these manually annotated and reviewed protein sequences are then fed back into a system run by one of the other teams at the EBI called Interpro. And this looks for sequence features for motifs and signatures which characterize groups of proteins. And they then assign, using, again, another expert curator, that those motifs and signatures are then used in these automated systems, and in particular in this one called UniRule. So it then identifies related proteins which share those motifs and signatures, and then the whole process begins again. getting onto a specific example now, exploring the function of this protein in UniProt. I've chosen a beta-lactamase as an example, and this particular one is called CTXM1. It's a class A beta-lactamase, and I'm going to guide you through the protein entry for this protein. This is what you would see on the website if you were to look for this particular protein. It's got a unique identifier that's here with this little gold icon next to it. That indicates that it's been manually curated. And then down the left-hand side here in this sidebar, this divides up the different sections of an entry. So you can click on any of these and then explore those sections separately. The other aspect I want to draw your attention to is that we refer to these as tabs. They're essentially headings. This is another way of viewing the data in the entry. So there's something called a feature viewer, which I'll say a bit more about later. The publications, which are the source of much of the data, the functional data associated with the protein. Links to external resources, and then a history. So, for example, if you wanted to know how this protein's entry has changed since it was first created, you can go work your way back from this tab, from this link. One other thing I wanted to highlight, and I put a red box around it, is that if you were to go and look at our public website now, you wouldn't actually see this. It doesn't go into the next update, unfortunately, until June. So at the moment, all you'd see is about, I think it's one sentence here instead of these 3 sentences, and also far fewer publications. Plus, it wouldn't have this section here, just in case you, out of curiosity, take a look for it. Again, working our way down a web page for this example, I've mentioned it's a beta-lactamase, so of course it's got a catalytic activity, and this is an example of some of the links which we have to other resources. So there's an EC or enzyme commission number, which is this, EC3526. There are the publications. Unfortunately, because this is from the current version, you can't see the publications. But you can see links to other resources. There's the enzyme database and Rhea. Rhea is a molecular function pathway, reaction database. It's a reaction database. So it would show something very similar to this, showing that if you took a beta-lactam, this is just a generic beta-lactam with the lactam ring here in the middle. And then these R groups are variable depending on which particular antibiotic we're talking about, how it gets hydrolyzed, and then converted into a substituted beta amino acid. Other information that's in an entry include keywords, other links to other enzyme and pathway databases like Brenda, a bit more information about where the names of the protein came from. These are some alternative names that this protein is known by. It's part of a whole group of caffeotaxamases. And this particular version of the protein was originally found in E. coli. I mentioned structures. This is how the structure is initially portrayed in an entry. The bar here, or the top row highlighted in yellow, is the highest resolution structure. So what we're seeing here is a representation of that structure. And you can see not only are there 4 published structures in the PDB database, there's also a predicted structure from AlphaFold. And you can get access to any of these from the entry. I mentioned the feature viewer. This is another way of viewing the data in an entry. And this one example I've chosen shows you the location of an active site in this beta-lactamase. It's, in fact, a serine residue. So, because it's a serine beta-lactamase. And there's a bit of information here about what goes on at the active site. It forms an acyl ester intermediate during the catalytic process. This is also something that will be updated in June. There'll be some papers added to this as experimental evidence that this is indeed the active site and it's the mechanism of action. And when you click on this particular residue in the feature viewer at the active site, the other thing that happens in synchronization with that is in the structure, you get a zoomed in view of the whole protein, and it highlights for you the specific residue at the active site. So this is the serine residue highlighted in green. I mentioned that it's part of the class A beta-lactamase family. This has been added by a curator manually. And there's a whole bunch of other family and domain database links here that you can use to follow up on other members of the family. Getting down now to the sequence itself. This is what the default view is in an entry. You can change this. You can also download it as in multiple formats. That's like the default one is the FASTA format, FASTA. And other sequence related information here. There's nucleotide sequences, protein sequences, which show you the origins, the original primary sequences, which were used to generate this protein sequence in UniProt when it was imported. Another view of related sequences is represented by this view. So these are similar proteins, and these have been pre-computed as proteins that match the protein of interest in your entry at different thresholds. So this is identical proteins. They're 100% identical, although they're not all necessarily the same length. If you notice down the side here, although they're identical, they don't match. Sorry, they're shorter. They're fragments, essentially, of the protein that's in the entry. But you can also see other proteins which match at different thresholds. Moving on to sequence analysis. Every entry is immediately accessible to do a sequence similarity search, a BLAST search with. And so there's a BLAST link in each entry, but there's also BLAST links on the homepage of the website. So you can get into BLAST from multiple directions. And this is what it would look like if I was to have clicked on that link in the entry. It will automatically put my sequence from that entry into the dialog box here. But if you wanted, you could choose to use a different identifier, the box at the top here. You can take any sequence succession that's present in UniProt, type it in here, and it'll appear here, and then you can run a BLAST search. There are various ways you can customize it or filter a search. You can choose which target database you search, and you can also restrict by taxonomy. So if you're only interested in bacteria or mammals or just mice or whatever, you can choose to put a name in here or a taxon ID if you know what it is. And this is what it would look like if I'd run a BLAST search with that sequence. This is the top, well, just a few of the 250 hits. I think the default is it gives you a maximum of 250. That might be adjustable, but I'm not quite sure. And having got this, you can look at the pairwise alignments of the hits in BLAST. So if I was to choose to look at this one here, which is called CTXM2, which is actually from Salmonella, this is what the alignment would look like. This loads up in the same window. And having done that, you might decide, well, actually, I want to see what some of these other proteins, related proteins, some of them quite similar, what their similarities and differences are. with my protein of interest. So I can run a multiple sequence alignment from this window. So if I'd use these checkboxes down the left-hand side, I can select, I don't know what the upper limit is. It might be something like 20, might be more. And then run a multiple sequence alignment. As you can see displayed in this window, this is how the dialog box would look. Here I've only got 2 displayed, but there are actually 6 in this multiple sequence alignment. And this is what it looks like when you look at the results. So I've highlighted here in this green box some of the things that you can do with a multiple sequence alignment. You can choose not just to look at similarity, but you can choose things like hydrophobicity and so on as a way of looking at the different parts of the alignment. And the other thing I've highlighted is if any of these proteins have got annotations associated with them, such as the presence of an active site, you can choose one of them as the source of the annotation, and then compare the set in the multiple sequence alignment and see just how well conserved a particular region or residue is. And in this case, you can see they've all got a serine at the same position for the active site. So that was a slightly rushed run through of an example of a protein entry, something that a beta-lactamase that's involved in AMR. What other things have we been doing, and in what other ways do we help AMR research? Well, I've got a list here of the different classes of proteins which we've considered as targets. So I've got at the top here, the most obvious ones perhaps are proteins involved in either drug degradation or transport. So one category of this is the beta-lactamases, and then also efflux pumps. And I'm going to say a little bit more about a couple of these secondary or indirect proteins that play an indirect role in AMR. Finally, I mustn't completely forget these 2 categories. Host proteins can behave as antimicrobials, as antibacterials, things like the defenses. And these could be used as models for potential drugs. And some of my colleagues are actually looking at these and also proteins encoded by bacteriophage. So the bacteriophage themselves can be used as a novel antimicrobial weapon. I've mentioned these 2 at the top, they're directly involved in AMR categories. So now I'm going to say a bit more about which beta-lactamases we've chosen to annotate. Now, all of them are very well annotated by automatic pipelines, but our manual annotation has been a bit patchy. And so we've chosen to focus on proteins which either have very few annotations already, or high clinical importance. And one of the ways well, this is going back to the example that I gave at the beginning, CTXM1. One of the ways in which we've improved the annotation for this particular entry is there's a better description of its function, describing the fact that it hydrolyzes both penicillins and cephalosporins, and that includes cefotaxime, which is quite an important cephalosporin antibiotic. The other thing that occurred when I was working on the beta-lactamases, I discovered that the workers in the field who knew more about it than I did had developed a novel or non-conventional amino acid numbering system. So I've included that in the entries now for the class A beta-lactamases. I was very confused for a while. So this gives you an idea of some of the groups of beta-lactamases which we've been annotating and either are in progress or we'll be getting to them soon. I've mentioned one of the members of the CTXM grouping. Actually, half a dozen of these have been annotated. And then these others, the GES and SHV families, are either moderately well covered or in progress. And we'll be getting to these others. And this is all based on a review that I found, which talked about clinical importance. That was one of the reasons these were chosen. Moving on to another example of a protein that's involved in drug transport. This is an efflux pump. It's a complex. It consists of 3 proteins in various stoichiometries. It was originally identified in Pseudomonas. In fact, it was the first multi-drug efflux pump identified in Pseudomonas aeruginosa. And we've improved its annotation by adding more papers from the literature, and also along in that process identified that it can be inhibited. The antibiotic efflux rather can be inhibited by this pyrimidine derivative, such as ABIPP. Moving on to other proteins which play a kind of indirect or secondary role in AMR, I'm going to give you one example of an ABC transporter. So these fall into 2 classes. They're either involved in acquiring and importing nutrients from the environment into a bacterium, or in turn they can do something a bit like the efflux pump that I mentioned before. They can export xenobiotics, and so that they may contribute to multi-drug resistance. And this is one which I worked on a little while ago. It's an interesting example because it's got mammalian orthologs. So it's not only found in these bacteria, in a whole range of pathogens, it's also found in mammals, or at least there's an ortholog of it in mammals. So we updated the papers associated with the entry for this protein. In fact, it's 2 proteins. It consists of the one in yellow here, and then these 2 in turquoise and blue. And one of these binds to the ligand, which is labeled here as QAC because it's a quaternary ammonium compound, but its proper name is ergothioneine. And it's something which many bacteria can't synthesize, hence they need to obtain it from the environment. Likewise, mammals can't synthesize it either, so we get it from our diet. So this is a novel ABC transporter, and the reason it's may play a role in AMR is that it is shown to be a virulence factor in Streptococcus pneumoniae, and it may also be involved in resistance against phagocytosis in MRSA. Finally, as an example, I'm going to just mention a protein which is not at all the kind of thing you pluck out of the air and say, oh, well, this plays a role in AMR. This is an aldehyde alcohol dehydrogenase. It's found in Streptococcus, but it's also got homologs in other species. And the group who studied, one of the groups who studied this protein, found out that it plays a role in enhancing virulence in mice. But what perhaps was most interesting was if one inactivates it, deletes it in the bacterium, it enhances susceptibility to antibiotics. And they also showed at the same time that rather than deleting it, there's this FDA-approved inhibitor of alcohol dehydrogenase, which is FDA-approved because it's used in humans. It also enhances susceptibility to antibiotics. So it could conceivably be thought of as an adjunct or an adjuvant to use in conjunction in the right circumstances with antibiotics. I mentioned the last section, I was going to say something about how does UniProt add value to the work of researchers other than what I've already described to you. And I just want to step through this. I've talked about manual curation of entries. Now, these annotations are used to propagate, is our technical term. Not really. When I first started with UniProt, I found it slightly confusing. Essentially, what it means is we make an evolutionary assumption that orthologs from closely related organisms are likely to have the same functions or very similar functions to those that we've demonstrated from experimental evidence from the literature. So we take annotations from those manually curated entries and propagate them to the orthologs. So things like their name, a brief outline, usually just a phrase about what the function of the protein is, if it has known catalytic activities, and so on. And these also act as templates for automatic annotation in the translated EMBL entries. Finally, all of our data gets disseminated very widely. It goes to other resources like Rhea, EnsemblBacteria, and then these other resources which are heavily used by the microbial, bacterial biology community. I think at that point, I might just skip through these last couple and sum up and allow a bit of time for any questions. So what I've done for you today is to introduce UniProt as a resource, say a little bit about how UniProt can help research in AMR, combines both manual and automatic annotations. I've taken you through some examples of proteins that we've annotated. And I'd like to just say, if you have suggestions for other proteins, other organisms that we should be focusing on in future, please let us know. And my last two slides, UniProt is a very big consortium. This just gives you an idea of the size of it. There are three different colors here. There's a group based in Geneva at the Swiss Institute of Bioinformatics. There's also a group in Washington, D.C. and Delaware, the PIR, and of course here at the EBI in Hingston. And so those colors, the red, blue and green, match up with the people here. And none of this is possible without funding. We have funding primarily from the National Institutes of Health, but also from other sources, EMBL itself, Open Targets, and the Swiss government through their CERI or CERI support for SIB. And so at that point, I'll stop and take questions.
Thank you very much, Paul. There is already one question, but I'd encourage people to write their questions into the Q&A if they have them now. Just before we have a look at those, I just wanted to mention about some upcoming webinars. So this today's webinar is part of an ongoing UniProt series, but we also have a microbial ecosystem series that is running on Wednesdays at the moment. There may be some interest for a couple of those for you coming up. So it's particularly one that's on the EnsemblBacteria genome browser that some of you may be interested in. So for questions, if you're able to pop those into the Q&A box so that we can kind of keep things in one place and keep track of them, that would be ideal. Okay, so let's have a look at those questions as they're coming through. So we have one here that is about a different organism. I know you were mentioning that at the end there. So it's asking about one specific organism, but perhaps you'll be able to share some ideas a bit more general. But it says, how will I be able to download AMPs in the black soldier fly in UniProt?
That's a specific. I can probably do it live. I could have a go.
A live demo.
Right. If I just go, for one thing, I don't know if. Let me try. Let's go to. Now, can you actually see? If I close that, can you see my web browser?
So no, we can still
No,
just see
no, you can still see the slides, aren't you?
Yeah.
Yes. So I'll have to stop sharing and then change to a different. Let me share this one there. Right. Hopefully now what you should be seeing is something called Swisslock.
It has not come through quite yet. The screen
oh, hang on, sorry.
Yes.
Now what you should be able to see is a UniProt entry.
That's correct. Yeah.
Yep. Okay. So, what was the name? Black.
So it's black soldier fly.
Black soldier fly.
That is also known as Hermetia illucens.
Well, let's see what happens if I just type black soldier fly. There's probably a better way of doing this, but Hermetia illucens.
Yes, that's the one.
That's the one. Okay. So there are ways in which you could focus it. So if I was to do an advanced search, I could add a field to this. So if I take let's just look at this. If I look at, there we are. That's so this is the entry, the taxonomic view in UniProt of Hermetia illucens, the black soldier fly. So if I now copy that taxon ID and go back to the view I had a moment ago and do an advanced search and then put taxonomy into here, that's now limiting my search to, so AMPs, antimicrobial proteins, peptides. I guess that's what they mean by AMPs. Well, if I try just AMP, no, let's try antimicrobial peptide. That could be what they mean. I can't spell. That's probably what they mean by AMP.
Yes, there's another comment to say yes, antimicrobial peptides.
Okay. So now I'm looking for the phrase antimicrobial peptide, but only in those species, that species. And it might be that there are other closely related species in UniProt, but that gives us a list there, quite a manageable list of 7. In fact, one of them is even reviewed. So this has been manually curated. Antimicrobial peptide active against gram-negative bacteria with a particular strain of bacteria, and not active against gram-positive bacteria. That's interesting. So there, that's how you could do it.
Great, thank you. I think that gives people an idea about the advanced search and restricting to specifics that they may have. Lovely. So that's the only question that we have in the Q&A at the moment. But as we mentioned earlier on, if you do have questions following this, you can absolutely get in contact with the UniProt team via the website, and they'll be able to help you to use the site. But we also have a number of different e-learning options on the EBI training site as well. I'll send a couple of links out with the recording so you can see some of that. But so, for example, we've had previous webinars that focus on the use of the API, which may be something that some of you are interested in using. So we'll send that out tomorrow. So I think that is all that we have on the questions front.
Okay.
Sorry, but obviously, as I say, this one more come through. Well, how do I search for MCR gene in UniProt? So again, I think this is
Okay.
Search.
I can do that. I think that means it's colistin resistance MCR. Okay, so if I try. Any organism. Okay, so if I was to go to that and go, I think the M in multiple, I think it stands for multiple. I've heard of the name MCR because there is a family of genes which have got that acronym in their gene name. So I could just try gene name multiple, and that doesn't find anything. Oh, that's because I'm sorry, I didn't change the taxonomy. Let's make it bacterial, shall we? So all bacteria, the taxonomic ID is 2. That doesn't find anything either. Okay. Let's try being more specific. And as I said, there are gene names which have got MCR in them, I'm sure.
The M is mobile.
Mobile. Yes. Yeah. Yes. So mobile. Yes, I think that's because many of them are encoded by plasmids or other mobile genetic elements. So yes, we can see here MCR family, something transferase. Yeah, there's a lot which have. One other thing you can do in a search is you can choose which information gets displayed. So I can choose, well, I'd like to see the gene name. We put gene name primary, and we go save. We can see, there we are, down the side here. We've got most of these, not all of them, have MCR in the gene name, which is what you'd hope from what I did in that search. So so mobile colistin resistance, that's how you could find one way to find proteins which are involved in that phenotype. Colistin's another antibiotic.
Great, thank you. Hopefully
Okay.
we're a little familiar now with the way the search works. We do actually have a few more questions now in the Q&A
Okay.
box. So we have here someone saying they use my pronunciation, I'm not sure about, but beta-lactamases as a pathway genome database. I think that's what it's saying, PDGB.
I think beta-lactamases is I'm not sure if we have links to beta-lactamases or not.
So saying I thought it was maybe on one of your slides, but
that was yeah. I go back. I can just remind myself here. So I thought it was EcoCyc. Oh. Just remember myself. Yeah, it's a related database that's maintained by the same people, I think. I talked about BioCyc and EcoCyc, which are a different class of data. So they hold information about bacterial genomes, whereas beta-lactamases is about pathways and metabolic pathways in particular. go back to there. So I don't think, do we have links to MetaCyc? I could find out. I might have to find that out tomorrow. But I can find that out.
So we can certainly get back to you on that one. And then I mean, obviously there's links to some other pathway databases, but obviously
yes, there are.
That one.
There are.
Okay. Okay. So there's a question about the alignment tool in UniProt and its reliability. So I think perhaps understanding that.
Yeah, I'm not sure how. I'm really not an expert on multiple sequence alignment for phylogeny, for a phylogenetic comparison. I'd have to ask one of my colleagues about that. I don't know, for example, which tools are built into UniProt to construct a phylogeny, because you can do that from a multiple sequence alignment. So yeah, if they would like to know an answer, I can find out from my colleagues.
So I think at this point, I just say thank you very much to Paul for his presentation today and for answering your questions. And thank you very much to all our attendees for coming along and joining us today.